Hi, good afternoon, everyone. A warm welcome to today's Fighting COVID-19 Asia Pacific webinar series by National Taiwan University. So I believe many of you have attended our last webinars where we discussed about the biomarker discovery using genomic platform. And today we will switch gear a little bit to talk about how the biomarker discovery also assists in therapeutic development for COVID-19. So big housekeeping rules. So if the attendees have any questions, please feel free to type your questions in the Q&A sections, or you can also chat with the host, which is me, Chen Qiao, and also our speakers. This webinar is going to be available on demand, so you can also feel free to view the webinar again if you want to learn more about the topics. So I would just like to review a little bit about NanoString's commitment to fight the COVID-19 pandemic. Since the beginning of the year, when the COVID-19 swept over the globe, NanoString had been dedicated to develop new gene expression tools where we can help the researchers in the field to profile the virus as well as a host response so that we can develop new tools, not only for vaccine, but also for drug treatments for the virus. So currently we have a few gene expression panels which have been widely used in the field, including the host response panels and also our immune and inflammation panels, where researchers can utilize to understand how the pathogens and host interaction can cause damage to the different organs. At the same time, we also develop new spatial biology tools, which is available on our Genomics DSP platform, where researchers can look into the virus really with the spatial context, meaning that they can see where the virus are. Are they in the lung? Are they in the heart? Are they in the liver? And where are all the immune cells interacting with this particular virus? And these spatial biology tools are not only developed by NanoString, but also together with our partners like ACD and also EPCAM. Since the beginning of the year, we have seen many publications on COVID-19. And here are just a few highlights of the COVID-19 publication where researchers utilize NanoString's technology on nCounter and Genomics DSP to understand the virus and the host response. As you can see here, that there are publications from New England Journal of Medicine, Science, Nature Medicine, and also journals that are not only focusing on the immune side of the virus, but also like the kidney damage, where the virus has been also causing problems for people with chronic disease. Lucky for us, today, we managed to invite one of our KOL in the field, who is Dr. Eugenia Ong. She is the one who lead the study for this particular publication, A Dynamic Immune Response Shapes COVID-19 Progression. This is a very special publication for Asia Pacific and particularly Southeast Asia region because it reports on the very first case of COVID-19 imported into Singapore. And later on, Dr. Ong will discuss about this study, her findings, and also some of the essays that she's currently working for the therapeutics and also the vaccine development. And again, this is a webinar series where we focus on the biomarker discovery, therapeutics, and also how we prevent the next wave or the next threat of pandemics. Now, for those who are interested, you can always view the webinars on demand and revisit the previous topics. And again, this is a collaboration between NanoString and National Taiwan University. With that, I would like to pass the webinar to Prof. Ruby Huang, who is the host and also the organizer for this webinar series. Prof. Huang.
Thank you everyone for tuning in for this webinar series. I'm Ruby Huang from National Taiwan University. So NTU, we have started this initiative of a research center for epidemic prevention science research, funded under the support of Ministry of Science and Technology in Taiwan, hoping to address not only this COVID-19 pandemics, but also to address how we accelerate the scientific research and international collaboration in the emerging infectious disease. So on behalf of NTU RECEPS as the the head of international collaboration. I thank you for your participation in this webinar series together with NanoString. Our purpose is to bring collaboration together to break the border boundaries, because we believe that we can only combat any pandemic, any epidemic like COVID-19 by working together. Thank you very much.
Thank you, Prof. Huang. Thank you very much. And now I would like to introduce our speakers for today's webinar. So the first speaker is Dr. Eugenia Ong from Duke-NUS Medical School. Dr. Ong is a Senior Research Fellow of Duke-NUS Medical School, and her expertise is in the immunology and also for infectious disease discoveries. So Dr. Ong's paper on the very first case of COVID-19 has struck a lot of interest from the field. And today we had the honor to invite her to share her expertise with our audience across Asia Pacific. And our next speaker after Dr. Ong will be Dr. Nicholas West. Dr. Nicholas West is research scientist from Griffith University in Australia. So Dr. West's expertise is in mucosal immunology. He's also currently the technology leader of the core lab facility in Griffith University, where he provides service and also a technology program to researchers across Asia Pacific. And this technology program, including pathology service, also encounter and genomics DSP service. So later, Dr. Nicholas West will also share his work on the COVID-19 and also how he can facilitate researchers across APAC to understand this pandemic better. Now with that, I will not delay any further, and I will pass today's webinar sharings to Dr. Ong. Hi, Dr. Ong, you can start sharing the slides now.
Okay. Hi, can you see the slides?
Yes, I can see the slide, Dr. Ong, and I can hear you very well.
Okay, great. So good afternoon, everyone. Thank you so much for attending the webinar today. First, I think I'd like to thank National Taiwan University as well as NanoString for organizing this webinar series, and really for the invitation for us to share our work with you today. So the topic that I'm going to be telling you guys all about today is our recent work on how a dynamic immune response shapes COVID-19 disease progression. And really this work was published in April as a brief report in Cell Host and Microbe. And so I think the distinctive feature of this work was that we had performed daily transcriptomic profiling on 3 COVID-19 cases. And as Chen Xiao had mentioned just now, they happened to be one of the first cases of COVID-19 that was imported in Singapore back in January of this year. So this includes one severe case, case 1 over here on the in blue, as well as 2 mild cases, case 2 and 3. And so really, the main takeaway from my talk today, if you don't remember anything else, is that the early immune response to COVID-19 is very dynamic. And through daily transcriptomic profiling, I think we were able to showcase dynamic nuances in the immune response, which you would otherwise not have captured without the intensive sampling. And so I think with more than 50 million cases to date and 1.3 million deaths worldwide, as of November 2020, I think SARS-CoV-2 has really resulted in a global health crisis. And we know that until an effective vaccine is widely available, understanding SARS-CoV-2 disease pathogenesis could lead to the development of host-directed therapies that could hopefully help to reduce the inflammatory response that we see as a hallmark of severe COVID-19. And so I think right at the start of the pandemic, the question on everyone's minds was whether or not the inflammatory response and cytokine were key drivers in COVID-19 severity. And so you'll see that in this figure that nicely summarizes both uninfected, moderate, as well as severe COVID-19, adapted from a review by Patterson and Ho, which was published in Journal of Clinical Investigation. If you look at the panel for severe COVID-19, you'll see that patients with severe COVID-19, they often have elevated levels of IL-6, TNF-alpha, as well as IL-10, so your classic pro-inflammatory mediators. In parallel, they also happen to have reduced CD4 and CD8 T cells, which I'll also later be sharing with you more about and about the potential roles of T cells in pathogenesis of SARS-CoV-2 infection. And so it seems that the inflammatory response is a hallmark of acute respiratory distress syndrome, or ARDS, as well as severe COVID-19. And so the question on our mind at that time was, indeed, how dynamic is this inflammatory response that we see in COVID-19 patients, and whether or not we know if this fluctuates extensively from one day to the next. And we thought that this was important because whether there is such a dynamic immune response, I think will be very important to studying COVID-19 pathogenesis. So especially in the context of defining the time from illness onset that needs to be accurately controlled when you have so many clinical studies in parallel looking at both vaccines and therapeutics for COVID-19. And so that was the question that we set out to answer. How dynamic is this inflammatory response? And so over here is the chronology of clinical features that we saw in our 3 COVID-19 cases that we profiled. So I'll go through this figure systematically. On the left-hand side, you'll see case 1 was a 66-year-old male. He was a tourist from Wuhan. Case 2 and 3 are the mild cases of COVID-19. Case 2 was a 37-year-old male, was the son of case 1, and case 3 was a 38-year-old male. So if you look at the timeline of clinical features for the 3 COVID-19 cases that have been profiled, you'll see that SARS-CoV-2 PCR from throat swabs was done on almost a daily basis in the first few patients that were admitted into Singapore General Hospital. And so a plus minus indicates positive and negative detection of the virus in throat swab, and a circle indicates that it wasn't done. So I think the distinguishing factor is that in case 1, who was the severe case, you'll notice that he was given supplemental oxygen of up to 4 liters over a course of 10 days. You'll also notice that in terms of the throat swab PCR, he was mostly PCR negative all the way up until he was fit for discharge, which is indicated by an asterisk on day 22. So in contrast, for cases 2 and 3, I think you'll notice that they mostly remain PCR positive for the most part of their stay in the hospital before they were eventually discharged roughly about a month later. So in terms of the clinical features of the 3 COVID-19 cases that we profiled, you'll notice that in case 1, which had more severe disease and also required oxygen supplementation, chest x-rays for case 1 showed the presence of bilateral patchy ill-defined lung infiltrates, as indicated by the arrows in red. So we only observed this in case 1, but not in case 2 or 3. So at the same time, we also looked at respiratory function in all 3 cases. And we looked at 3 different factors. So if I could orientate you to the graph that you see over here on the screen, on the x-axis, we have days post-illness onset. And for our y-axis, we have oxygen saturation, which is in blue, supplemental oxygen, which is in pink, and beats per minute or heart rate, which is in purple. So you'll see that despite being given 4 liters of supplemental oxygen at around day 5 to 7 post-illness onset, case 1 still had a fairly low oxygen saturation of around 94%. And this was also right around where heart rate was very high. And so we inferred that respiratory function was actually most compromised on around day 5 post-illness onset in case 1. And so to really address the inflammatory as well as immune response to SARS-CoV-2 infection, especially at very early stage of the disease, and to find out whether that is indeed a dynamic response, we profiled transcriptomic changes in the blood using the Encounter Human Immunology panel. And I think what we found was that there was a highly dynamic expression of pro-inflammatory genes in our COVID-19 cases. And so if I could just orientate you to this heat map that you see on the screen. So this heat map was generated from unsupervised hierarchical clustering of gene expression after nanostring profiling. So on the rows, we have the genes, and in columns, we have our samples. So the gene expression levels, red correspond to high expression levels and blue corresponds to lower expression levels. So we had healthy controls that were included in this analysis, and this is indicated, the labels are in yellow on the left-hand side of the heat map. For our 3 COVID-19 cases, they are to the left, to the right of the heat map, and below the labels for the COVID-19 cases, you'll also notice that the days from illness onset are indicated below. So what we found was that as compared to healthy controls, the gene expression profiles that we are seeing in COVID-19 cases, they look extremely different. And we were able to, based on the hierarchical clustering, split the genes up into 4 distinct clusters. Genes in each cluster were then put through a pathway enrichment analysis, and we've annotated the respective clusters with the most significant pathways that were driving the gene expression that you see over here on the heat map. So if I could turn your attention to cluster 4, I think you'll notice that many of these genes that are driving toll-like receptor as well as inflammatory signaling, they are peaking at day 6 post-illness onset in case 1, and that's actually peaking after the nadir of respiratory function, which we had shown just now was at day 5. And so this included cytokines like IL-2, IL-6, and therefore we questioned the cytokine storm hypothesis when we saw the data coming up. So in contrast, I think you'll see that there's a reversal in the trend in cluster 3, where we see that instead now in case 1, we have reduced levels of genes that are present in cluster 3, and these are genes that are mainly driving antigen presentation. And it also included T-cell markers such as CD4, CD8A, and CD8B, which I'll go on to show you the data for later on. And so the expression for these genes were actually downregulated in case 1 as opposed to both healthy controls as well as the mild cases, case 2 and case 3. So for a start, we immediately kind of just zoomed in on pro-inflammatory cytokines that were known to drive potentially cytokine storm, as well as other pro-inflammatory factors that were known to correlate with respiratory distress. And however, when we looked at the expression of cytokines such as IL-2, IL-6, and TNF-alpha, we saw that the expression is actually either within the range of the mild cases or healthy controls. So healthy controls on the graph that you see is indicated with a dotted line for the mean, and the gray shaded areas represent the standard deviation. So you'll see that the expression of IL-2, IL-6, and TNF-alpha, really it's within the range of mild cases or healthy controls. So interestingly, what we found was that only expression of the cytokines IL-1A, 1B preceded the nadir of respiratory function. And so although these findings come from a single subject, I think it also raised the possibility that increased expression of IL-1 as well as its associated signaling pathway could be responsible for driving COVID-19 pathogenesis. And in parallel, what we saw were that the T-cell markers CD4, CD8A, as well as CD8B, there was reduced expression in case 1 as compared to both healthy controls or case 2 and case 3, which were mild cases. And we thought that it could suggest recruitment of T cells from the peripheral blood to the site of infection or to draining lymph nodes, which could have led to the mostly negative SARS-CoV-2 RT-PCR findings that we had saw in the throat swabs in case 1. And so in contrast, we think that the reduced pro-inflammatory as well as the attenuated T cell activation that we are seeing in case 2 and 3 could have resulted in milder symptoms, but led to an extended PCR positivity that we saw in both case 2 and case 3. So in any case, at that time, I think it suggested to us that T cells really have a role to play in the pathogenesis of the disease. And I think a lot of other groups have gone on to show that indeed T cells are important. We're also saying for T cell mediated immunity seems to be very important for vaccine-related protection. And so with our findings, I think there were 2 possibilities that came up with what we had found from the nanostring profiling that was done on whole blood from COVID-19 patients. The first is that if you have reduced activation of adaptive immune genes, I think that could actually lead to a potentiation of increase in toll-like receptor inflammatory signaling, and this could happen in a positive feedback loop, as had been demonstrated by Kim and colleagues in the Nature paper in 2007. Also, in addition, the increased levels of pro-inflammatory mediators in case 1, I think, could have led to recruitment of CD4 as well as CD8 T cells from peripheral blood to sites of inflammation and draining lymph nodes. And that could have resulted in the mostly negative throat swab RT-PCRs that we had seen in case 1. And so really, with the gene expression profiling that we had done, I think one of the initial motivations was also, once we could understand how dynamic this immune response was, whether we could then apply host-directed therapies to perhaps bring down the level of inflammation that we saw, especially in patients with severe COVID-19. And I think indeed, there's already been a Lancet Rheumatology paper in which they had used off-label anakinra, which is a recombinant IL-1 receptor antagonist, and they found that it reduced the need for mechanical ventilation in ICU as well as mortality among patients with severe COVID-19. However, of course, confirmation of efficacy will still require randomized controlled trials. So in a setting that allows you to really dissect the efficacy properly. So I think in summary, our findings hint at the potential of IL-1 as well as the related pro-inflammatory pathways as a potential therapeutic target for COVID-19. It also suggests an important role for T cells in COVID-19, as we saw that attenuated cytokine and T cell activation in mild infections could actually lead to prolonging of infection. And so I think defining the role, especially of T cells in either pathogenesis of severe COVID-19 or prolonged infection, could also help to inform case management or as well as virus transmission control. And this work especially, I think, highlights the dynamic inflammatory and immune responses to SARS-CoV-2, and hopefully it also guides the design of future studies to understand COVID-19 pathogenesis. And so with that, I'd like to acknowledge Viremix, my colleagues at Viremix, especially my bosses, Eng-Yong and Jenny, for giving me the opportunity to work on the COVID-19 studies, our colleagues at Duke-NUS Medical School, as well as Singapore General Hospital. We thank all doctors and nurses in the isolation facility in Singapore General Hospital, and Viremix was established through a generous support from the Tanoto Foundation. Thank you so much for your attention.
Thank you, Dr. Ong. Thank you so much for your generous sharing on this research paper. I just had a couple of very curious questions before I go to the audience to see if anyone had any questions. So just now you had showcased that the anti-IL-1, the IL-1 antagonist could be a potential therapeutics for these patients. So my first question is that I actually seen some other paper mentioning maybe anti-IL-6 and some other cytokine antagonists could be used for treating the patient who are severely ill. Do you foresee this kind of antagonists for the cytokine will be used as a single reagent, or do you needs to be used in a combined cocktail format? Because most of the time we are seeing the cytokine storm had a couple of factors increasing concurrently. So this is actually my first question. And the second question is really about the treatments that you have identified signatures and genes that will continue, that will be able to hopefully segregate the patients. My question is, do you see that these treatments will have a dose-dependent effect, or do they have to further, do we have to further categorize the patient into different subgroups and give them different treatment? For example, certain patients will receive only IL-1, or certain patients who are more severely ill will actually receive a cocktail of these this antagonist, of cytokine antagonist.
Right. So thanks, Tian-Ta, for your question. So these are actually really important questions that I think we're also currently trying to work out. I don't think there is any correct answer. Okay, so for the first question that you had on the use of the anti-cytokine treatments as a way of bringing down the inflammatory response, I think you are right. There have been multiple studies that have that are actively in progress. If you look at clinical trials that are currently being done, there are probably a few for both IL-6 as well as IL-1 kind of inhibitors. I think the key thing is that in planning some of these studies, one of the things that we think about is also whether or not the patients are treated early enough, because from the gene expressions profiling that we had done, it seems that a lot of these pro-inflammatory factors, they actually peak at very early time points following infection. And so I think it is also logistically difficult in terms of when you are recruiting patients into such clinical trials, and you may not be able to standardize when patients are receiving certain treatments. And so that's when I think randomized clinical trials, a randomized controlled clinical trial is really important, because then you are able to standardize when patients are receiving treatment versus maybe a placebo. And that really, I think, informs the analysis, and it reduces a lot of other confounding factors, right? So, and then on your second question, where you had asked about, I think the gene signatures related to COVID-19 and what we're seeing in patients. So based on the work that we've done, so over here I've showed you the gene expression profiling from one severe case as well as 2 mild cases. But I think right now there's a whole lot of information that's actually has been already published in the last few months, because that information is coming out daily, and it's great because it really adds to the breadth of knowledge that we are starting to get on the pathology of the disease, on the immune response following infection, and so on. And it is possible, I think, that through such a method, so probably just not our data set alone, but if you were able to kind of be able to look across the wealth of data that has already been published, I do think that it is possible in terms of if you are able to compare time points across the board for patient samples, to be able to do things like patient stratification and to look at whether or not there could be dose-dependent changes depending on the treatment being given. But again, I think it goes back to that question again of the sampling process that's being done. So when we started this study, I think we were very clear in that we wanted to perform intensive sampling, and so we did that instead of focusing on a larger number of patients. So it allowed us to get a higher resolution at how the gene expression changes are differing on a day-to-day basis. And I think with that wealth of knowledge, we also have to take into account how the sampling was done for certain studies. And hopefully, if you're able to put all that data together, you are able to get a very specific, maybe COVID-19 signature that could predispose, that you could then identify and maybe which we think could then predispose someone to more severe disease, or whether you think someone might just, have mild disease and so on. Sorry, I think my answer was quite long.
But thank you very much on the sampling, and also the particular early response when the patient was first taken to the. And that actually leads us to the next questions. This question is from Prof. Ruby Huang. She wondered if the serial gene expression profiling that you have shown just now, are they being routinely done in all the COVID-19 patients in Singapore, or they are more really for, I mean, this is more for myself. I wonder if there's more for those who are recruited into clinical trials. Is there a routine that we will do serial gene expression profiling now?
So actually, I think most of the time this is not done because the intensive sampling, I think, requires quite a lot of coordination between both the clinical site as well as the lab. And I think we were able to do this because we have a team that has strengths in both areas. So my PI, Dr. Jenny Loh, she's a senior infectious disease consultant at SGH, and she's really the one who has been supporting a lot of all these clinical studies on the side of the ward. And I think, just other than the science, there is a lot of background coordination that needs to happen. A lot of logistical details need to be worked out ahead of time. So the answer is no. In fact, this type of routine intensive sampling is not really performed, not for clinical trials. We did it because I think at the beginning, we really wanted to understand whether or not that, how the immune response actually differs from day to day, which we thought could then help to inform on some of the host-directed therapies that were currently being floated. So yeah, the answer is no.
Thank you very much for that. But what we can see from just a study for that is actually quite a lot of parties to be come together to contribute to this study, and we want to thank you. Really, in the early days of the COVID pandemic, that you basically are risking your own health to perform all this study with the case when we basically know nothing about the virus back then. So thank you very much for the contribution.
Thank you.
And so now, in the interest of time, we will actually move to our next session. And as many of the audience already know, that currently there is this vaccine and drug treatment race in the world to develop new drugs and also new vaccine treatments for the COVID-19 pandemics. And for example, for Dr. Ong's study, we are also looking out to, in the future, we can collect all the researchers in APAC to come together to form a network where we can work together towards this goal. Now, in order to do this, sometimes for this kind of cross-border collaboration, we do need a very dedicated team who great expertise in genomic studies, and also not just in the bulk tissue genomic study, but also specialized in certain more advanced technology like spatial biology platforms. So this will bring us to our next speaker, Dr. Nicholas West, who is the research scientist from Griffith University. So Dr. West is currently leading the Center of Excellence of Spatial Biology in Griffith University. And thank you, Dr. West, for sharing your slides already. So please start your session sharing. Thank you.
Thank you, QQ, and hello, everybody. Nice to e-meet you. I think these webinar series are excellent as a format for us to be able to share information, particularly across borders when we can no longer travel. As QQ mentioned, my talk is going to focus on two aspects. One is our Centre of Excellence in Spatial Biology program, and the second will be to just provide a brief update on some immune profiling that we have done here on the Gold Coast in COVID-19. There we go. So the first thing I just wanted to quickly mention is that Griffith University has been working with NanoString now to develop a Centre of Excellence in Spatial Biology, following the long history we've got looking at genomics research. Our research group has now got a 10 or 15 year history looking at genomics, particularly in obesity, oncology, and type 2 inflammation, particularly around eczema and allergy. We decided to have a look at the spatial biology platform, the genomics platform with NanoString, based on early work that was being done to show just how well it characterized immune cells and molecules within the tissue environment. You can see here on the left-hand side that I've got an image of the genomics platform. So we've been very fortunate now to be working with NanoString and the platform for the last 6 months to really optimize the processes to efficiently be able to analyze samples. We've got our own work in oncology, type 2 inflammation, and in infectious disease. We've also got some COVID-19 research, but we've also been working with many institutional partners and with commercial partners who have been collecting tissue in their own research programs. Our own philosophy is that we're keen to be able to generate data and help people with their research. We work with groups to prepare their slides. We then also run the samples, run the samples and provide you with a report on your data. And you can see some of the main areas that we have been able to do research in. The oncology program is at this stage by far the leading program, with work in multiple myeloma, lymphoma, immunotherapy, and in radiation oncology. We've now been looking at a second machine to allow us to ensure that we can maintain quick turnaround times, and we've got projects that range from 2 slides out now to 112 slides, so they're relatively big programs. For groups that might be interested in accessing the service that we provide, our work is part of the Systems Biology and Data Science group at Griffith University, and I've put our email or our website address down here at the bottom. And so if anyone is interested, you can email me. My email is n.west@griffith.edu, and I'll go through that again at the end of the presentation. Or you can go through our website and it will have information on the work that we do to try and help people generate data. Beyond that, I wanted to then follow up on the great talk that we just saw looking at immune profiling in COVID-19 to look at some work that we've done here on the Gold Coast. For anyone who doesn't know, Australia has been very successful in controlling the spread of COVID-19. We've had one major outbreak in Victoria, and we've currently got an outbreak in South Australia. But otherwise, similar to Singapore, we've been able to very much control the spread of COVID-19. But there is strong interest in being able to understand exactly what it is that separates those people who have a mild response or an asymptomatic response from those who have a critical response and who then end up in the intensive care unit, escalates to respiratory failure and potentially death. So there's been a lot of interest in trying to understand the immune response. And the slide that you've got here in front of you really is the generic immune response to a virus, where you can see an early phase response here, followed by some cellular immune responses, particularly a lymphopenia. There is somewhat of an anemia seen in most viral responses as the immune system generates an antibody response. But for COVID-19, a lot of different treatments have been tried, standard virology treatments, and those haven't been particularly successful until recently. So the research that we wanted to have a look at was to quantify changes in mucosal tissue and systemic viral RNA during the course of COVID-19 illness. And the tissue component was very important for us, given spatial biology. We wanted to have a look at characterising whole blood tissue and mucosal inflammatory responses to SARS-CoV-2 using the nCounter and Geomics digital immune gene expression profiling platforms. We wanted to also have a look at tissue and whole blood immune responses to SARS-CoV-2 using mass cytometry. And we wanted to examine the relationship between mucosal tissue and systemic immune responses with SARS-CoV-2 viral load and with clinical symptoms and health outcomes. So this was a fairly broad project. Our aim was to really understand what happens in the mucosa, what happens in the nasopharyngeal swabs. So we were able to collect nasopharyngeal swabs from people when they were diagnosed. All people who on the Gold Coast who were diagnosed with coronavirus had to come into hospital for an initial checkup. We would then collect a blood sample as well. And as this diagram shows, we were then able to collect samples periodically through the course of infection. So we had at admission and diagnosis, the collection of blood and a nasopharyngeal swab. 48 to 96 hours post-diagnosis, we would again try and collect the same samples. Most people at this point were discharged from hospital, and so we were able to collect samples here again. But for those people that needed admission to ICU, escalation of respiratory support, or ICU discharge, we were able to collect a full set of samples. And then for those people who progressed to respiratory failure, we looked to collect a lung, liver, and renal sample. I'll quickly work through some results for you, and we saw some of this in our last speaker's talk. This is a heat map that shows the expression of immune genes in the mucosa. So this is the nasopharyngeal swab. And this looks at the 770 gene pan-cancer immune profiling panel. And you can see here, we had 2 patients that were critically ill and one who was admitted to ICU, but not that sick, who didn't require respiratory support. And we then had 3 asymptomatic individuals. And you can see here through an unsupervised hierarchical cluster that we had our 2 critically ill patients with quite high immune gene expression throughout the whole of their mucosa. Compare that to our 3 asymptomatic patients, and you can see a very starkly different gene expression profile. And then we had our moderate patient who really did sit outside the other 2. And I think the most interesting area is this set of genes here that we can see were upregulated in our critically ill, were not so upregulated in our asymptomatic individuals, but we then have this mixed profile in our moderately ill person. And from this heat map, I only pulled out 2 genes. This is immune regulatory factor 2 and immune regulatory factor 5. And the interesting thing you'll notice here is that despite our critically ill patients here having this broad upregulation of their immune genes, the virus-specific immune genes, antiviral genes, their expression was higher in our asymptomatic group compared to our symptomatic group. So this to us was very interesting to see that key antiviral immune genes were upregulated in our asymptomatic individuals. When we then had a look at their whole blood immune gene expression profile, again using the pan-cancer immune profiling panel, we saw a very similar pattern of immune gene expression. And I'll just draw your attention to the top here, because a lot of people might say, well, that looks exactly the same, but I'll go back a slide and you'll see here that this set of genes up here were not really impacted in the mucosa. So this is definitely a different slide, just for those people who might be wondering. But you can see here in an unsupervised cluster again, that we have our critically ill patients here who had an upregulation of pretty much all their immune genes in blood. We had our asymptomatic individuals here with this very much lower immune gene expression profile, and our moderately ill person here who separated out somewhat differently. When we then had a look at a volcano plot, you can see here that this is comparing symptomatic to asymptomatic. So all the genes that are out on this side of the volcano plot are increased in expression in our symptomatic individuals, with the most highly expressed gene, this interferon alpha receptor 2 gene. And this is one of the key genes that is upregulated in a coronavirus and a viral infection. So to some extent, you'd expect to see these genes upregulated, but interestingly that this one was so highly significantly expressed in our symptomatic individuals. Perhaps the thing that most interested us was this panel down here, which is the immune cell abundance score. So the immune cell abundance scores look at the set of immune genes in a cell and then deconvolutes that to give an immune cell score. And you can see here that in our asymptomatic individuals, we had a much stronger immune response, in particular a T helper 1 or cell-mediated immune response. So you can see our T cells were higher in our asymptomatic individuals, Th1 cell scores, CD8 T cell scores, our macrophage cell scores, and our B cell score, which is obviously important then for our antibody response. Interestingly, our NK cell score showed the opposite response. So what we've seen in both the mucosa and in blood is that we've got a general upregulation across the immune system in our symptomatic individuals, but in relation to specific antiviral responses, we're starting to see very, very specific patterns that our asymptomatic individuals are perhaps able to mount a more specific antiviral response to coronavirus. This diagram down here is a KEGG pathway diagram, and this allows you to see the expression of genes on their pathway map, their biological function. And you can see here in orange, we've got our genes that were upregulated in our symptomatic group compared to our asymptomatic group. So this just allows us to have a look on a biological level at what genes are doing in a pathway. And this is a fairly canonical immune pathway, the JAK-STAT signaling pathway. So to finish up with some concluding comments, it appears that in our research in COVID-19, our asymptomatic individuals have a more focused antiviral response in the mucosa. And then in the systemic immune profiling, our asymptomatic individuals have a more robust cell-mediated immune response. We're continuing to undertake analysis of samples, particularly our tissue samples, which we've only just finished getting ethics approval for. And we've been impacted by our low caseload. So unfortunately here on the Gold Coast, we've only had about 190 individuals. Most of those have been in quarantine. And so we are continuing to collect samples from our colleagues from across the country. The final picture I wanted to show is of our research group, and this is just at our Christmas party last year. This is the group of researchers that are now running our Centre of Excellence. We're led by Professor Alan Cripps, who has a long history in vaccines and in mucosal responses. Particularly want to note Yelena Vida and Amanda Cox for their work in the NanoString Centre of Excellence, and Ping Zhang, who is our bioinformatician. So we're able to offer people both the sample processing assistance, but also bioinformatics and data science. And we do have some initiatives having a look at the analysis of images that are generated on the spatial profiling platform. Thanks very much, QQ.
Thank you, Dr. West. Thank you very much for your sharing on your Center of Excellence and how you do the immune profiling in COVID-19. And that's actually a very interesting case that where we see you are profiling both the mucosal samples and also a blood sample. I mean, from the previous talk where Dr. Ong has sharing that most of the profiling has been done in the whole blood. But in this case, we see that you are shifting some of your samples to a mucosal sample. And you also mentioned that some of the sample potentially will be profiled at a tissue level.
Yes.
So that is really great to see that we are getting researchers in different fields to understanding the COVID-19 biologies at different level. I do have questions regarding your Center of Excellence, the Spatial Biology program. So first of all, congratulations on the low case of COVID-19 infection in your region, which probably is a good problem not to have so many cases in your region. But we do know that you are also not getting enough sample for much broader studies. So currently, are you looking into getting imported samples, not imported case, imported samples from other parts of the world? For example, in United States or maybe even South America or India, where we can see that the daily cases is still at 100K or even 200K. Are you looking into doing some collaboration with the researchers in this part? And on top of that, we know these are highly infectious samples, right? So how do you think you can control the cross-border study, like for example, in your Center of Excellence, or like Dr. Ong's study in Singapore, if there was something collaboration come together, what will be your recommendation to handle such infectious sample, maybe in the future, not just for COVID itself, but maybe for other infectious agents that we can research on?
Yes, that's a great question, QQ. Australia has actually directives that COVID-19 samples can be handled at PC2 level. And so that just requires standard biological transport requirements. So we're able to ship samples internationally. And in fact, we do have one COVID collaboration in the US where we're collecting samples and receiving, or they're collecting samples there and we're receiving them. We would be very happy to look at collaborating either in a pure service agreement where we can help run the samples, or in a more collaborative arrangement where we're able to assist with the research questions. So that's certainly no problems on our end. We have been talking potentially to groups in Southeast Asia, but as yet we don't have any formalised agreement. So we would certainly be interested in talking to people. We handle all samples as per standard biological controls. We have a university biosafety committee. Most of our samples, we've actually tested our COVID samples for viremia, particularly blood, and we haven't been able to detect any at all. So at this point, standard fixation techniques are all that are required for the transport and handling of samples.
Thank you, Dr. West. Thank you so much on this information. So we had another questions regarding the mucosal and also the blood sampling. And so in this study, you had focus on both sample type. So Prof. Ruby Huang wonders, like, do you think in the field, for those who are already running in clinical trials, they should consider running pair samples for both blood and mucosal samples in the clinical setting? Or do you think that this will still remain something more on the research end to understand the biology of the virus?
So I guess that's a difficult question. The primary interests for our research were twofold. One is, can we identify patients who are going to progress to severe disease? And what can we identify that might be used in drug repurposing? I think the question about nasopharyngeal swabs and early decisions for treatment is important. At this stage, we think that there is a difference between asymptomatic individuals and symptomatic individuals in the mucosal control, and that's most likely to do with the innate immune system. In actual fact, your interferon, regulatory genes, interferon-related factors, your NOD proteins. At this point, we think it is still worthwhile doing both nasopharyngeal swabs and saliva samples along with blood samples, because that provides a full picture as to what's going on. By the time you see an adaptive immune response or the lack of a response, and particularly for elderly people where many people don't mount a secondary antibody response because their T follicular helper cell pool is now depleted. So that's one of the standard issues with vaccine responses, and being able to identifying someone when they've got disease is almost too late. We do have a delayed time. It's day 10 or 11. In fact, at the Gold Coast, it was 11.8 days that we saw people either progress or recover. And so there is somewhat of a short time frame to determine whether you want to escalate responses. But by that stage, I think that probably the disease processes are now well underway, and you want to be able to get that earlier. So potentially it would seem that decision may be made if you could understand the mucosal immune responses in nasopharyngeal swabs.
Thank you very much for the explanation. So I think for today's webinar series, we had Dr. Ong, who has shared about the very first case from Singapore on the COVID-19 disease dynamic, and then we had Dr. West, who has shared on the mucosal and also the blood samples and on the COVID-19 testing. So for those who had questions for our speakers, please feel free to email them if you want to discuss more, particularly for future collaboration. And as Dr. West has shared with the audience, that currently Griffith University provides a service program and also technology program for the spatial biology as the Center of Excellence. And if you are interested, I welcome everybody to join the virtual launch of the Center of Excellence tomorrow at Griffith University from 2 to 3 p.m. Singapore, so 12 to 1 p.m. Singapore time, which will be 2 to 3 p.m. Brisbane time or Gold Coast time. This is going to be an online meeting launch. Unfortunately, because of the pandemic travel ban, we can't come together. But if you are interested, please feel free to join us at this virtual launch. And I just put the registration link in the chat group where you can sign up. And we will have more sharing on the Griffith University's program, the technology program, on how it can assist people not just in COVID-19, but also in other cancer immunology. So with that, I would like to thank both our speakers and Prof. Ruby Huang for organizing this amazing webinars again. This webinar is again available for on-demand viewing, and please feel free to come back to us to ask more questions. And Prof. Huang, thank you very much. I would like to invite you for a closing remarks.
Thank you everyone to join the second part of the webinar series. Thank you, Duke-NUS, for helping with this fantastic organization, and thank you, Nick, and then Eugenia, for bringing us this information on how the profiling could be done on the serial manner or on mucosal profiling, and then that definitely would open up a lot of new opportunities for cross-border collaborations. Thank you again, and we still have the third webinar series coming up. Please do sign up.
Thank you, Prof. Huang. So in this case, I will keep the room, the webinar room, open for another 2 minutes for anyone who had any questions or who want to reach out to Dr. West and Dr. Ong if you want to discuss more in details for collaborations. And in 2 minutes time, I will close the webinar and I will see you in our third part of the COVID-19 webinar series. Thank you very much. Thank you, everyone. We will close the webinar room now.